The other day I was talking to my neighbor, a retired biology teacher, and she mentioned something that completely blew my mind. She said that her granddaughter, who's studying genetics at college, told her that scientists can now edit human DNA like we edit a Word document, cut, copy, paste, delete. But instead of fixing typos in an essay, they're fixing typos in the very blueprint of life itself. At first, I thought she was exaggerating, but then I started digging into this technology called CRISPR, and honestly, what I discovered changed my entire perspective on what's possible in medicine and science today. Just like finding that amazing wine for $39 that turned out to have a 4.2 rating on Bovino. Sometimes the most revolutionary discoveries come from the most unexpected places. CRISPR didn't start in some high-tech laboratory with scientists in white coats trying to cure disease. It started with bacteria, tiny microscopic organisms that have been fighting viruses for billions of years, developing their own sophisticated immune system that makes our human immune system look like child's play. Back in 1987, a Japanese scientist named Yoshizumi Ishino was studying bacteria when he noticed something strange in their DNA. There were these weird repeating sequences that didn't seem to serve any purpose. For decades, scientists largely ignored these sequences, dismissing them as junk DNA. But just like that wine representative who told me about the same bottle being sold for $300 at a gourmet store, sometimes what looks ordinary on the surface contains extraordinary value underneath. It wasn't until 2005 that researchers began to understand what these DNA sequences actually did. A Spanish scientist named Francisco Mojica was the first to realize that bacteria were essentially keeping a genetic photo album of every virus that had ever attacked them. When a virus invades a bacterium, the bacterium doesn't just fight it off. It takes a little piece of the virus's DNA and stores it in these special sections called CRISPR-As. It's like keeping mugshots of every criminal who's ever tried to rob your house. But here's where it gets really fascinating. When the same type of virus attacks again, the bacterium doesn't have to start from scratch. It pulls out the mugshot, makes copies of it, and uses those copies to guide molecular scissors called Cas proteins to the exact location of the viral DNA. The Cas proteins then cut up the virus's genetic material, destroying the invader before it can cause damage. It's a search and destroy system that's more precise than anything we've ever built. The breakthrough moment came in 2012 when two women, Jennifer Doudna from UC Berkeley and Emmanuel Charpentier, then at the University of Vienna, figured out how to reprogram this bacterial immune system. They realized they could design synthetic guide molecules that would direct the molecular scissors to cut any piece of DNA they wanted. It was like taking a heat-seeking missile designed to hit one specific target and reprogramming it to hit whatever target you choose. What made this discovery so revolutionary wasn't just that it worked, but how simple it was compared to previous gene editing tools before CRISPR. Editing genes was like trying to perform surgery with a chainsaw while blindfolded. It took months or years to modify a single gene, cost hundreds of thousands of dollars, and often didn't work at all. CRISPR changed that overnight. Suddenly, labs all over the world could edit genes in a matter of days for a few $100s. It democratized genetic engineering in a way that nobody had anticipated. The first hint of CRISPR's medical potential came through the story of Victoria Gray, a 34-year-old woman from Mississippi who had suffered from sickle cell disease her entire life. Victoria's story is heartbreaking in its simplicity. Her red blood cells, which should be smooth and round like donuts, were twisted into painful sickle shapes because of a single letter change in her DNA. Just one letter out of the 3 billion letters in the human genome, but that one mistake had tormented her for decades. Victoria had spent her entire adult life in and out of hospitals, experiencing what doctors call vaso-occlusive crises, episodes where her misshapen blood cells would get stuck in her blood vessels like cars in a traffic jam, causing excruciating pain that could last for days or weeks. She tried every treatment available, taken every pain medication prescribed, but nothing could address the root cause of her suffering. In July 2019, Victoria became the first person in the United States to receive CRISPR gene therapy for sickle cell disease. The process was complex and risky. Doctors extracted millions of stem cells from her bone marrow, used CRISPR to edit those cells in the laboratory, and then destroyed her existing bone marrow with chemotherapy before infusing the edited cells back into her body. It was essentially a bone marrow transplant using her own cells, but cells that had been genetically modified to produce healthy hemoglobin. The waiting period was agonizing. Victoria had to endure months of uncertainty, not knowing whether the treatment would work or whether she had just subjected herself to dangerous chemotherapy for nothing. But 8 months after the treatment, something remarkable happened. Victoria woke up one morning and realized she felt different. For the first time in her adult life, she wasn't in pain. Today, more than 5 years later, Victoria Gray is completely free from sickle cell crises. She works full-time, travels internationally as a speaker, and has energy to fully engage with her for children in ways she never could before. When reporters ask her to describe the difference, she says it's like the difference between night and day. The treatment didn't just improve her condition, it appears to have cured her completely. Victoria's success wasn't a fluke. By the time the Food and Drug Administration approved the therapy called Casgevy in December 2023, clinical trials had treated 4 to 4 patients with sickle cell disease. Of the 31 patients who had been followed long enough to evaluate the treatment's effectiveness, 29 were completely free from pain crises for at least 12 consecutive months. That's a success rate of over 93% for a disease that was previously considered incurable. But sickle cell disease was just the beginning. The same CRISPR therapy that cured Victoria Gray also works for beta thalassemia, another blood disorder that requires patients to receive blood transfusions every few weeks for their entire lives. In the clinical trials, 25 out of 27 patients with beta thalassemia no longer needed transfusions after receiving CRISPR therapy. The two patients who still needed transfusions saw their transfusion requirements drop by 80 and 96% respectively. The approval of Casgevy marked a historic moment, the first time the FDA had ever approved a therapy based on CRISPR technology. But even as this milestone was being celebrated, researchers were already working on the next generation of applications that would make CRISPR even more powerful and precise. One of the most exciting developments has been the integration of artificial intelligence with CRISPR technology. Remember how I mentioned that CRISPR works like molecular scissors guided by a genetic GPS system? Well, artificial intelligence is making that GPS system incredibly more accurate. Traditional methods of designing guide RNA letters, the molecules that tell CRISPR where to cut, were often hit or miss. Scientists would design several different guides and hope that at least one would work well. Now, AI systems like Deep CRISPR can predict with over 97% accuracy which guide RNA letters will work best for any given target. Machine learning algorithms analyze millions of previous CRISPR experiments to understand the subtle patterns that determine success or failure. It's like having a GPS system that not only knows every road but can predict traffic conditions, construction delays, and the best route before you even start your journey. But perhaps the most remarkable AI breakthrough came in 2024 with the development of OpenCRISPR-1, the world's first entirely artificial gene editor. A company called Profluent Bio trained AI models on over 5 million naturally occurring Cas proteins from across the tree of life. The AI learned the rules that govern how these molecular scissors work, and then designed completely new proteins that had never existed in nature. OpenCRISPR-1 isn't just slightly better than natural CRISPR systems, it's dramatically more precise, with a 95% reduction in off-target effects compared to the standard Cas9 protein. It's like the difference between a scalpel and a butter knife. This artificial protein is over 400 mutations away from any natural Cas protein, meaning it's genuinely a new form of life designed entirely by artificial intelligence. The precision improvements are crucial because one of the biggest concerns about CRISPR has always been the possibility of off-target effects, unintended cuts in the genome, that could potentially cause harm. Early CRISPR systems occasionally cut DNA at the wrong locations, creating mutations that could theoretically lead to cancer or other problems. While these off-target effects were relatively rare, they were still a significant safety concern, especially for therapeutic applications. The development of high-fidelity CRISPR variants combined with I-design guide RNA letters has reduced off-target effects to nearly undetectable levels. Modern CRISPR systems are so precise that they can distinguish between DNA sequences that differ by just a single letter. It's like being able to edit one specific word in a library of books without accidentally changing any other words that look similar. This precision has opened the door to treating genetic diseases that were previously considered too risky to target. Take Leber congenital amaurosis, a form of inherited blindness caused by mutations in a gene called SEPT9O. The eye is actually an ideal target for CRISPR therapy because it's relatively isolated from the rest of the body and because small improvements in vision can have enormous impacts on quality of life. Companies are now testing CRISPR therapies that are injected directly into the eye to correct the genetic defects that cause blindness. Cardiovascular disease represents another frontier where CRISPR is showing tremendous promise. A company called Verve Therapeutics is using base editing, a variant of CRISPR that can change single letters of DNA without cutting both strands to target the PCSK9 gene in the liver. People who are born with natural mutations that reduce PCSK9 function have dramatically lower cholesterol levels and reduced risk of heart disease throughout their lives. The Verve therapy aims to recreate these protective mutations in people who weren't born with them. Early results from clinical trials show that a single injection can reduce LDL cholesterol, the bad cholesterol, by 30 to 50% for what appears to be years, or possibly decades. If successful, this approach could prevent heart attacks and strokes in millions of people with a one-time treatment. Cancer treatment has been another area where CRISPR is making remarkable progress, particularly through CAR T-cell therapy. Traditional CAR T therapy involves extracting a patient's immune cells, genetically modifying them to better recognize and attack cancer cells, and then reinfusing them back into the patient. The process has shown remarkable success against certain blood cancers, but traditional methods of genetic modification have limitations. CRISPR is supercharging CAR T therapy by allowing multiple precise edits to be made simultaneously. Researchers can knock out genes that make T cells become exhausted, insert new genes that help them persist longer in the body, and remove the natural T cell receptor to prevent graft versus host disease. These armored CAR T cells are showing improved effectiveness and persistence compared to conventional CAR T cells. One of the most exciting developments is the creation of off-the-shelf CAR T cells using CRISPR, instead of using each patient's own T cells, which can take weeks to manufacture and cost hundreds of thousands of dollars. Companies are creating universal CAR T cells from healthy donors. These cells are edited with CRISPR to remove the markers that would normally cause them to be rejected by the patient's immune system. The result is a standardized cancer treatment that could be available immediately when needed, rather than requiring weeks of manufacturing time. The success of these early CRISPR therapies has created enormous excitement in the medical community, but it has also raised some difficult questions about access and equity. Casgevy, the approved CRISPR therapy for sickle cell disease, cost about $2.2 million per patient. While this might seem shocking, it's actually comparable to other gene therapies and potentially cost-effective when compared to a lifetime of medical care for sickle cell disease. But the high price creates a cruel irony. Sickle cell disease predominantly affects people of African descent, communities that have historically faced significant barriers to accessing advanced medical care. The disease is most common in sub-Saharan Africa, where millions of people suffer from sickle cell but have limited access to basic medical care, let alone cutting-edge gene therapy. The manufacturing requirements for current CRISPR therapies create additional barriers to access. The process requires sophisticated cell processing facilities, highly trained technicians, and months of treatment time. Patients must travel to specialized centers, often far from home, and undergo intensive medical procedures including chemotherapy. There are only about 200 bone marrow transplant centers in the entire United States, and only 3 in all of sub-Saharan Africa. However, innovative solutions are beginning to emerge. Canada has developed a point-of-care manufacturing approach that produces CAR T cells for about $40,000, compared to $475,000 for commercial products. If this approach can be scaled and adapted for CRISPR therapies, it could dramatically reduce cost and increase accessibility. The development of in vivo CRISPR therapies treatments that deliver gene editing directly to cells inside the patient's body rather than editing cells outside the body could also improve access. These therapies would eliminate the need for cell extraction and processing, potentially making CRISPR treatment as simple as receiving an injection. Several companies are making progress on in vivo applications. Intellia Therapeutics has developed treatments for hereditary transthyretin amyloidosis and hereditary angioedema that use lipid nanoparticles to deliver CRISPR directly to the liver. These treatments have shown remarkable success in early trials, with single injections providing years of benefit. The success of CRISPR in treating individual genetic diseases has also sparked interest in more ambitious applications. Agricultural uses of CRISPR are addressing global food security challenges by creating crops that are more nutritious, climate resilient, and productive. Like traditional genetic modification, which often involves inserting genes from other species, CRISPR can make precise changes that are indistinguishable from natural mutations. Researchers have used CRISPR to create wheat varieties that are resistant to drought, rice that produces higher yields, and tomatoes with improved nutritional content. The technology can also reduce food waste by creating fruits and vegetables that don't brown or spoil as quickly. These applications can be crucial for feeding a growing global population in the face of climate change. But perhaps the most ambitious CRISPR projects involve what scientists call gene drives, genetic modifications designed to spread through entire wild populations. The most advanced applications target disease-carrying mosquitoes, with the goal of reducing malaria transmission. Researchers have developed CRISPR-modified mosquitoes that are unable to carry the malaria parasite, or that produce offspring that are predominantly male, which could cause mosquito populations to crash. Gene drives represent both enormous promise and significant risk. On one hand, they could eliminate diseases that kill hundreds of thousands of people annually. On the other hand, releasing genetically modified organisms into the wild could have unpredictable ecological consequences. The technology is so powerful that it has sparked international discussions about governance and oversight. The ethical questions surrounding CRISPR become even more complex when we consider the possibility of editing human embryos while somatic editing, modifying the cells of living patients, is widely accepted for treating serious diseases. Germline editing, making changes that would be inherited by future generations, remains highly controversial. In 2018, a Chinese scientist named He Jiankui shocked the world by announcing that he had created the first gene-edited babies. He used CRISPR to disable the CCR5 gene in human embryos, with the goal of making the resulting children resistant to HIV infection. The announcement was met with global condemnation from the scientific community, and he was subsequently imprisoned. The He Jiankui incident highlighted the need for international governance of CRISPR technology. Currently, 75 out of 96 countries surveyed have policies prohibiting heritable human genome editing, with most having outright bans. No country explicitly permits germline editing, reflecting global consensus that the technology isn't ready for such applications. However, the scientific capability to edit human embryos raises profound questions about the future of human evolution. If CRISPR technology becomes safe and precise enough, should we use it to prevent genetic diseases before children are born? Where do we draw the line between treating disease and enhancing human capabilities? These questions become even more complex when we consider applications like improving disease resistance, enhancing cognitive abilities, or modifying physical characteristics. The integration of CRISPR with other emerging technologies promises even more dramatic capabilities. Researchers are combining gene editing with stem cell technology to create patient-specific tissues and organs for transplantation. Others are using CRISPR to engineer microorganisms that can produce pharmaceuticals, biofuels, or other valuable compounds. The convergence of CRISPR with artificial intelligence is creating possibilities that seemed like science fiction just a few years ago. AI systems are now being used to design entirely new biological systems, predict the effects of genetic changes, and optimize gene editing strategies in real time. This combination of technologies is accelerating the pace of discovery and making previously impossible applications feasible. Looking ahead, the next decade promises to be transformative for CRISPR technology. We're likely to see approvals for dozens of additional gene editing therapies, targeting everything from inherited blindness to neurodegenerative diseases. Manufacturing innovations will make treatments more accessible and affordable. New delivery methods will enable treatment of previously untargetable diseases. The story of CRISPR is ultimately a story about human ingenuity and the unexpected places where breakthrough discoveries can come from. Who would have thought that studying the immune systems of bacteria would lead to treatments that can cure genetic diseases that have plagued humanity for thousands of years? It's reminiscent of how that simple wine I found at the supermarket opened my eyes to the complexities and hidden values in an entire industry. Just as the wine industry has had to grapple with questions about authenticity, value, and accessibility, the CRISPR revolution is forcing us to confront fundamental questions about what it means to be human, who should have access to genetic enhancement technologies, and how we can ensure that the benefits of this powerful technology are shared equitably around the world. The patients like Victoria Gray who have benefited from CRISPR therapy are living proof of the technology's transformative potential, but they're also pioneers who took enormous risks to access experimental treatments. Their courage is paving the way for millions of future patients who will benefit from safer, more effective therapies. As we stand on the brink of a new era in medicine, it's important to remember that CRISPR is not just a scientific tool, it's a reflection of our highest aspiration as a species. The desire to eliminate suffering, to cure disease, and to improve the human condition has driven every major advance in medicine throughout history. CRISPR gives us unprecedented power to achieve these goals, but with that power comes unprecedented responsibility. The choices we make today about how to develop, regulate, and deploy CRISPR technology will shape the future of medicine and human civilization for generations to come. We have the opportunity to create a world where genetic diseases are a thing of the past, where personalized medicine is available to everyone, and where the accidents of birth no longer determine a person's health and potential. But realizing this vision will require more than just scientific breakthroughs. It will require thoughtful governance, international cooperation, and a commitment to ensuring that the benefits of genetic medicine are shared by all of humanity, not just those who can afford to pay for them. The story of CRISPR is still being written, and all of us have a role to play in determining how it ends. What amazes me most about CRISPR is how it demonstrates that the most profound discoveries often come from the most unexpected places. Those bacterial immune systems that seemed like biological curiosities turned out to hold the key to revolutionizing medicine. It makes me wonder what other extraordinary discoveries are hiding in plain sight, waiting for someone curious enough to look closer and ask the right questions. The future of CRISPR is limited only by our imagination and our wisdom in wielding this incredible power. The technology that started with studying bacteria fighting viruses has already transformed medicine and is poised to reshape agriculture, environmental conservation, and perhaps even human evolution itself. It's a reminder that science, at its best, doesn't just answer questions, it opens up entirely new possibilities we never knew existed. And that, in the end, might be the most remarkable thing about CRISPR. It's not just a tool for editing genes, it's a tool for editing the future itself. But let me tell you about some of the other incredible stories that are unfolding right now in CRISPR research. Remember how I mentioned that Victoria Gray was the first patient with sickle cell disease to receive CRISPR therapy? Well, there's another patient named Alyssa who represents a completely different application of this technology. Alyssa was a 13-year-old girl from Leicester, England, who had T-cell acute lymphoblastic leukemia that didn't respond to any conventional treatments. By the time doctors suggested CRISPR therapy, she had already undergone chemotherapy, radiotherapy, and a bone marrow transplant. But nothing had worked. Alyssa received what's called CAR T-cell therapy, but with a CRISPR twist. Instead of using her own immune cells, which were compromised by her leukemia, doctors used T-cells from a healthy donor and edited them with CRISPR to create what they called off-the-shelf CAR T-cells. These donor cells were modified to avoid rejection while specifically targeting her cancer cells. The process required precise CRISPR editing to knock out the genes that would normally cause graft-versus-host disease while preserving the cells' ability to fight cancer. 10 months after receiving the CRISPR-edited CAR T-cells, Alyssa's cancer was undetectable. She's now back in school, playing with friends, and living a normal teenage life. Her case represents the first successful use of CRISPR-edited donor cells to treat cancer, opening up possibilities for treating patients who can't wait weeks for their own cells to be modified. The CAR T field is exploding with innovation thanks to CRISPR. Traditional CAR T therapy has shown remarkable success against certain blood cancers, with cure rates approaching 80% for some types of lymphoma. But the process has limitations. It takes for weeks to manufacture each patient's customized CAR T cells, and about 10 to 20% of patients experience severe side effects from the treatment. CRISPR is addressing both of these challenges. Companies like Allogene Therapeutics and Selectus are creating universal CAR T cells that can be manufactured in large batches and stored frozen until needed. These cells are edited with CRISPR to remove the HLA proteins that would normally cause rejection, making them invisible to the patient's immune system. The result is a cancer treatment that could be available off the shelf like any other medication, but researchers are going even further. They're using CRISPR to create what they call armored CAR T cells with multiple enhancements. They can knock out the PD-1 gene. To prevent the cells from being turned off by tumor defenses, they can insert genes for cytokines that help the T cells proliferate and persist longer in the body. They can remove the T cell receptor to prevent graft versus host disease, while preserving cancer-fighting ability. Some of the most exciting recent results come from trials of CRISPR-enhanced CAR T cells targeting CD19, a protein found on certain blood cancer cells, in one trial conducted by Caribou Biosciences. 15 out of 16 patients with aggressive non-Hodgkin's lymphoma responded to treatment, with 7 achieving complete remission lasting 6 months or longer. These response rates are comparable to or better than conventional CAR T therapy, but with the potential for faster manufacturing and lower costs. The success in blood cancers is driving efforts to apply CRISPR-enhanced CAR T therapy to solid tumors, which have been much more challenging to treat. Solid tumors create hostile environments that can exhaust T cells and prevent them from reaching cancer cells. But CRISPR is enabling researchers to engineer T cells that are more resistant to these hostile conditions. One approach involves editing T cells to resist the immunosuppressive signals that tumors use to protect themselves. Another involves modifying T cells to produce enzymes that can break down the physical barriers that surround solid tumors. Researchers are also engineering T cells to target multiple cancer antigens simultaneously, making it harder for tumors to escape by losing a single target protein. The applications extend beyond cancer. Researchers are using CRISPR to create therapeutic cells for autoimmune diseases, metabolic disorders, and even neurological conditions. One particularly exciting application involves engineering regulatory T cells, specialized immune cells that suppress inflammatory responses to treat autoimmune diseases, like multiple sclerosis and type 1 diabetes. The precision of modern CRISPR systems has also enabled the development of what's called base editing and prime editing. These are variants of CRISPR that can make precise changes to DNA without creating the double-strand breaks that were a hallmark of the original technology. Base editing can change single letters of DNA, while prime editing can make small insertions, deletions, and replacements with incredible precision. These newer editing tools are particularly important for treating genetic diseases caused by point mutations, single-letter changes in the DNA that can have devastating effects. About 60% of known genetic diseases are caused by point mutations, making base editing and prime editing potentially applicable to thousands of different conditions. Prime Medicine, a company founded by David Liu who invented prime editing, is advancing the first prime editing therapy toward clinical trials. Their lead program targets chronic granulomatous disease, a rare immune deficiency caused by mutations in genes required for immune cell function. Prime editing can precisely correct these mutations without causing the DNA breaks that could lead to unwanted side effects. The delivery challenge has been one of the biggest obstacles to expanding CRISPR applications beyond blood cells that can be edited outside the body. Getting CRISPR into the right cells inside a living patient requires solving complex problems of targeting, uptake, and avoiding immune responses to the delivery vehicles. Lipid nanoparticles, similar to those used in COVID-19 vaccines, have emerged as one of the most promising delivery methods. These microscopic fat bubbles can encapsulate CRISPR components and deliver them to specific tissues. The key breakthrough came with the development of tissue-specific lipid nanoparticles that preferentially accumulate in the liver, lungs, or other target organs. Intellia Therapeutics has been a leader in developing in vivo CRISPR therapies using lipid nanoparticles. Their treatment for hereditary transthyretin amyloidosis (HATTR) uses CRISPR to knock out the TTR gene in liver cells, preventing production of the misfolded proteins that cause this devastating disease. In clinical trials, a single injection has provided sustained reductions in disease-causing proteins for more than a year. The results have been remarkable. Patients with HATTR typically experience progressive nerve damage and heart problems that can be fatal within a few years of diagnosis. But patients who received Intellia's CRISPR therapy have shown sustained improvements in disease biomarkers and quality of life measures. The treatment appears to halt disease progression and may even reverse some of the damage. Similar approaches are being tested for other liver diseases. CRISPR can knock out genes that cause hereditary hypercholesterolemia, potentially providing permanent reductions in cholesterol levels. It can target genes involved in hemophilia, potentially eliminating the need for lifelong clotting factor injections. It can modify genes involved in metabolic diseases like primary hyperoxaluria, which causes kidney stones and kidney failure. The eye represents another attractive target for CRISPR delivery because it's relatively isolated from the rest of the body and because even small improvements in vision can be life-changing. Editas Medicine has been developing a CRISPR therapy for Leber congenital amaurosis 10, a form of inherited blindness caused by mutations in the CEP290 gene. The treatment involves injecting CRISPR components directly into the eye, where they can edit the genetic defect in light-sensing cells. Early results from clinical trials have been encouraging, with some patients showing improvements in light sensitivity and visual function. The therapy targets a specific mutation that affects about 10 to 20% of patients with Leber congenital amaurosis, but success could pave the way for treatments targeting other genetic causes of blindness. Researchers are also making progress on delivering CRISPR to the brain and nervous system, despite the challenges posed by the blood-brain barrier. The viral vectors that can cross this barrier are being developed to deliver CRISPR for neurological diseases. Some of the most promising applications target single-gene neurological disorders, like Huntington's disease, where knocking out the mutant gene could halt disease progression. The agricultural applications of CRISPR are equally transformative, and perhaps even more far-reaching in their potential impact. Like the medical applications we've been discussing, which affect individual patients, agricultural CRISPR could impact billions of people by improving food security, nutrition, and environmental sustainability. The first CRISPR-edited crops are already reaching consumers in some parts of the world. In Japan, you can buy tomatoes with enhanced levels of GABA, an amino acid that may help reduce blood pressure. The tomatoes were created using CRISPR to knock out a gene that breaks down GABA, allowing the compound to accumulate to levels 5 times higher than in conventional tomatoes. In the United States, several CRISPR-edited crops have been approved by regulatory agencies and are making their way to market. These include soybeans with improved oil profiles, corn with enhanced nutrient content, and wheat varieties with improved yield characteristics. Because many of these crops contain only deletions or modifications that could occur naturally, they don't require the extensive regulatory approval processes that traditional genetically modified crops undergo. But the most exciting agricultural applications of CRISPR are still in development. Researchers are creating crops that can thrive in challenging environments. Created by climate change, drought-tolerant varieties of rice, wheat, and corn could help maintain food production as rainfall patterns become more unpredictable. Salt-tolerant crops could allow farming in areas where sea level rise has contaminated soil with salt water. CRISPR is also being used to improve the nutritional content of staple crops, golden rice, which has been genetically modified to produce. Vitamin A has been in development for decades but faced regulatory and technical challenges. CRISPR offers a more precise approach to nutritional enhancement that can overcome some of these obstacles. Researchers have used CRISPR to create rice varieties with enhanced iron and zinc content, addressing micronutrient deficiencies that affect billions of people worldwide. They've developed wheat varieties with improved protein quality and reduced gluten content for people with celiac disease. They've created cassava varieties with enhanced vitamin content and reduced levels of naturally occurring toxins. Disease resistance is another major focus of agricultural CRISPR research. Plant diseases cause crop losses worth billions of dollars annually and threaten food security in many parts of the world. CRISPR can be used to knock out genes that pathogens use to infect plants, or to enhance plants' natural immune responses. One of the most promising applications involves citrus greening disease, which threatens to devastate the global citrus industry. The disease is caused by bacteria that are spread by insects and has already destroyed millions of citrus trees in Florida and other growing regions. Researchers are using CRISPR to create citrus varieties that are resistant to the bacteria, potentially saving an industry worth billions of dollars. The livestock applications of CRISPR are equally impressive. Researchers have created pigs that are resistant to porcini reproductive and respiratory syndrome, a viral disease that costs the swine industry billions of dollars annually. They've developed cattle with improved disease resistance and heat tolerance, traits that will become increasingly important as climate change affects animal agriculture. But perhaps the most ambitious agricultural application of CRISPR involves gene drives, genetic modifications designed to spread through wild populations. The most advanced applications target mosquitoes that carry diseases like malaria, dengue fever, and Zika virus. These diseases affect hundreds of millions of people annually and cause enormous suffering in tropical and subtropical regions. Oxitec, a British biotechnology company, has developed genetically modified Aedes aegypti mosquitoes that carry a gene drive designed to suppress wild mosquito populations. The modified mosquitoes produce offspring that die before reaching maturity, causing population crashes when released into the wild. Field trials in the Cayman Islands and Brazil have shown significant reductions in mosquito populations in release areas. But gene drives represent both enormous promise and significant risk. Unlike conventional genetic modifications that affect only the organisms that receive them, gene drives are designed to spread through entire populations over multiple generations. This means that a single release could potentially affect mosquito populations across vast geographic areas. The ecological implications are profound and largely unknown. Mosquitoes play important roles in many ecosystems as pollinators and food sources for other animals. Eliminating or drastically reducing mosquito populations can have cascading effects throughout food webs. There's also the possibility that gene drives could spread beyond their intended targets or evolve in unexpected ways. Recognizing these risks, researchers are developing safeguards for gene drive technology. These include self-limiting gene drives that are designed to disappear from populations after a certain number of generations, and reversal drives that could be used to counteract unwanted effects. Some researchers are also exploring the use of gene drives to modify mosquito populations rather than eliminate them, such as making them unable to carry disease-causing pathogens. The regulatory and ethical challenges surrounding gene drives are enormous. because released gene drives can potentially cross national borders and affect shared ecosystems. They require international coordination and governance. The Convention on Biological Diversity has called for extensive risk assessment and stakeholder engagement before any environmental releases of gene drive organisms. The convergence of CRISPR with artificial intelligence is accelerating progress across all these applications. AI systems can analyze vast amounts of genomic data to identify the best targets for gene editing, predict the effects of specific modifications, and optimize guide RNA designs for maximum effectiveness and minimal off-target effects. Machine learning algorithms are being used to predict which genetic modifications will produce desired traits in crops, animals, and microorganisms. This computational approach can dramatically reduce the time and cost of developing new applications by allowing researchers to test thousands of different modifications virtually before conducting expensive and time-consuming laboratory experiments. AI is also improving the safety of CRISPR applications. Deep learning models can predict potential off-target effects with increasing accuracy, allowing researchers to design guide RNA letters that minimize unwanted cuts. Other AI systems can analyze the effects of gene edits on cellular metabolism, protein function, and organism development to predict potential unintended consequences. The combination of AI and CRISPR is enabling the development of entirely new biological systems that don't exist in nature. Researchers are using AI to design synthetic biological circuits that can perform complex computational tasks inside living cells. These engineered systems could be used to produce pharmaceuticals, detect diseases, or even terraform other planets. One particularly exciting application involves using AI-designed CRISPR systems to create living sensors that can detect environmental pollutants, pathogens, or chemical weapons. These biological sensors could be more sensitive and specific than conventional detection methods, while being completely biodegradable and environmentally safe. The manufacturing and delivery challenges that currently limit CRISPR applications are also being addressed through innovative approaches. Point-of-care manufacturing systems could bring gene editing treatments to underserved populations by eliminating the need for centralized production facilities. Automated cell processing systems could reduce cost and increase consistency of cellular therapies. Alternative delivery methods are expanding the range of treatable diseases. Researchers are developing implantable devices that can produce and release CRISPR components over extended periods, potentially enabling treatment of chronic diseases with periodic gene editing. Others are exploring the use of modified bacteria or engineered microorganisms as living delivery vehicles that can target specific tissues or disease sites. The cost considerations that currently limit access to CRISPR therapies are beginning to be addressed through innovative pricing models and manufacturing improvements. Value-based pricing tied to treatment outcomes can make expensive therapies more accessible by spreading cost over time based on effectiveness. Subscription models similar to those used for hepatitis C treatments could provide unlimited access to gene editing therapies for fixed annual fees. International initiatives are working to bring CRISPR treatments to underserved populations. The World Health Organization has established advisory committees on genome editing to develop global standards and promote equitable access. Several foundations are funding research specifically aimed at developing low-cost CRISPR treatments for diseases that predominantly affect people in developing countries. The regulatory landscape for CRISPR continues to evolve as agencies worldwide grapple with the unique challenges posed by gene editing technologies. The FDA has established specialized review pathways for gene and cell therapies that can accelerate approval of breakthrough treatments while maintaining safety standards. European agencies are developing similar frameworks that balance innovation with precaution, but regulatory harmonization remains a significant challenge. Different countries have different standards for safety, efficacy, and ethical acceptability of gene editing applications. This fragmentation can slow development of new treatments and create inequities in access to approved therapies. The intellectual property landscape surrounding CRISPR is complex and continues to evolve. Patent disputes between major institutions have created uncertainty about who owns the fundamental rights to CRISPR technology. These disputes could affect the availability and cost of future treatments. Though many companies are working around potential patent issues by developing alternative editing systems, the ethical considerations surrounding CRISPR applications continue to evolve as the technology advances. While there's broad consensus that using gene editing to treat serious diseases in consenting patients is ethically acceptable, questions remain about enhancement applications, germline editing, and environmental releases of modified organisms. Public engagement and education are crucial for ensuring that CRISPR technology develops in ways that are aligned with societal values and priorities. Surveys consistently show that public support for gene editing varies significantly, depending on the specific application, with strong support for treating serious diseases but more skepticism about enhancement or agricultural applications. The international scientific community has established several mechanisms for ongoing dialogue about CRISPR ethics and governance. The International Summit on Human Genome Editing brings together scientists, ethicists, policymakers, and patient advocates to discuss emerging issues and develop consensus recommendations. Professional organizations have established ethics committees and guidelines for responsible conduct of gene editing research. Looking toward the future, Several trends are likely to shape the development of CRISPR technology over the next decade. The integration with AI will continue to accelerate, making gene editing more precise, predictable, and accessible. New delivery methods will enable treatment of previously untargetable diseases and tissues. Manufacturing innovations will reduce costs and increase access. The applications will continue to expand beyond current medical and agricultural uses. Environmental applications could include using CRISPR to clean up pollution, restore damaged ecosystems, or adapt species to changing climate conditions. Industrial applications could involve engineering microorganisms to produce sustainable materials, fuels, or chemicals. The convergence of CRISPR with other emerging technologies will create entirely new possibilities. Combining gene editing with synthetic biology could enable the creation of entirely artificial organisms, designed for specific purposes. Integration with nanotechnology could enable precise delivery of gene editing treatments to individual cells. Combination with regenerative medicine could enable the creation of replacement organs, tailored to individual patients. But perhaps the most profound impact of CRISPR will be on our understanding of life itself, by giving us the ability to systematically modify genes and observe the effects. CRISPR is accelerating our understanding of how genes function, how diseases develop, and how biological systems work. This knowledge is already leading to new therapeutic targets and treatment approaches that don't involve gene editing. The democratization of gene editing enabled by CRISPR is also changing the landscape of biological research. Small companies and academic laboratories can now tackle problems that previously required the resources of major pharmaceutical companies. This democratization is accelerating innovation and enabling research on rare diseases and other areas that might not be commercially attractive to large companies. The story of CRISPR demonstrates the unpredictable nature of scientific discovery. The researchers who first studied bacterial immune systems couldn't have imagined that their work would lead to treatments for genetic diseases, tools for improving crops, or methods for combating infectious diseases. This unpredictability underscores the importance of supporting basic research, even when the applications aren't immediately obvious. As we stand on the brink of this new era in biology and medicine, it's worth reflecting on the responsibilities that come with such powerful tools. CRISPR gives us unprecedented ability to modify living organisms. But with that ability comes the obligation to use it wisely. The choices we make about how to develop, regulate, and deploy gene editing technologies will have consequences that extend far beyond our own lifetimes. The patients like Victoria, Gray, and Alyssa who have already benefited from CRISPR therapies are living proof of the technology's transformative potential. But they're also reminders that behind every scientific breakthrough are real people whose lives hang in the balance. The urgency of their need for effective treatments must be balanced against the importance of ensuring safety and considering long-term consequences. The success stories we've seen so far represent just the beginning of what's possible with CRISPR technology. As the tools become more precise, the delivery methods more effective, and our understanding of biology more complete, the range of treatable conditions will continue to expand. Diseases that seem incurable today may be as treatable as infections are now. But realizing this potential will require continued investment in research, thoughtful regulation that balances innovation with safety, and ongoing dialogue about the ethical implications of genetic modification. It will require international cooperation to ensure that the benefits of gene editing are shared globally rather than concentrated in wealthy countries. Most importantly, it will require maintaining focus on the ultimate goal, reducing human suffering and improving the quality of life for people around the world. The wine industry analogy I started with seems almost trivial in comparison to the stakes involved with CRISPR, but the underlying principle remains the same. Sometimes what appears ordinary on the surface contains extraordinary value underneath. The bacterial immune systems that seemed like scientific curiosities turned out to hold the key to revolutionizing medicine. The question is, what other revolutionary discoveries are waiting to be uncovered in the vast unexplored territories of biology? As CRISPR technology continues to evolve and mature, we can expect to see applications that we can barely imagine today. The tools that allow us to edit genes may eventually allow us to edit aging, disease susceptibility, and perhaps even human capabilities themselves. The boundary between therapy and enhancement will continue to blur as our understanding of genetics improves and our technical capabilities expand. The story of CRISPR is ultimately a story about human potential, both the potential to discover incredible new tools and the potential to use those tools to create a better world. Whether we realize that potential will depend on the choices we make as individuals, communities, and nations. The technology itself is morally neutral. It's how we choose to use it that will determine whether CRISPR becomes a force for healing and improvement or a source of inequality and unintended consequences. What gives me hope is the dedication and wisdom I've seen from the researchers, clinicians, and patients who are pioneering these applications. From the scientists who spent decades studying bacterial immune systems to the patients who volunteer for experimental treatments. The CRISPR revolution is built on a foundation of curiosity, courage, and compassion. If we can maintain those values as the technology continues to advance, there's no limit to what we might accomplish in the decades ahead. The momentum in CRISPR research shows no signs of slowing down. Just in the past few months, we've seen remarkable breakthroughs that would have seemed impossible just a few years ago. Take the work being done on primary hyperoxaluria type 1, a devastating inherited disorder that causes kidney damage through the overproduction of oxalate. Biotech Therapeutics recently reported that their in vivo CRISPR therapy reduced harmful oxalate levels by nearly 70% in patients after a single treatment. The therapy uses lipid nanoparticles to deliver CRISPR directly to liver cells, where it deactivates the gene responsible for oxalate production. What makes this particularly exciting is that it represents a new frontier in CRISPR applications, treating metabolic disorders with single injections that can provide years or decades of benefit. The traditional approach to managing primary hyperoxaluria involves strict dietary restrictions, medications, and often kidney transplantation. But this CRISPR therapy could potentially eliminate the underlying cause of the disease with a one-time treatment. Another breakthrough came from Precision Biosciences, which reported encouraging initial data for their treatment of chronic hepatitis B virus infection. This is particularly significant because current hepatitis B medications can suppress the virus, but can't eliminate it completely. The virus creates what doctors call covalently closed circular DNA in liver cells, essentially a permanent reservoir that allows the infection to persist for life. But Precision's CRISPR therapy aims to cut out these viral reservoirs completely, potentially providing a true cure for a disease that affects over 190 million people worldwide. The early results are promising. 2 out of 3 patients showed substantial reductions in viral markers, even at the lowest dose tested. If successful, this approach could eliminate the need for lifelong antiviral medications and remove the constant threat of liver damage that hepatitis B patients face. These advances in liver-targeted therapies are part of a broader trend toward in vivo CRISPR applications that can treat diseases with simple injections, rather than complex cell processing procedures. The liver has emerged as an ideal target because it's easily accessible through the bloodstream. It's responsible for producing many disease-causing proteins, and it has a remarkable ability to regenerate after genetic modifications. But researchers aren't stopping with liver diseases. Recent breakthroughs in delivery technology are opening up possibilities for treating diseases in virtually every organ system. Scientists have developed tissue-specific lipid nanoparticles that can preferentially deliver CRISPR to muscles, lungs, brain, eyes, and other organs. Each breakthrough in delivery expands the range of treatable diseases exponentially. The neurological applications are particularly exciting because they address some of humanity's most devastating conditions. Huntington's disease, a genetic disorder that causes progressive breakdown of nerve cells in the brain, has been a prime target for CRISPR research. The disease is caused by a toxic mutant protein, and researchers have shown that using CRISPR to reduce production of this protein can halt disease progression in animal models. But delivering CRISPR to the brain presents unique challenges. The blood-brain barrier, which normally protects the brain from toxins and infections, also blocks most therapeutic molecules from reaching brain cells. However, researchers have developed modified viral vectors that can cross this barrier in the liver, CRISPR specifically to affected neurons. Early trials for Huntington's disease are expected to begin within the next few years. Amyotrophic lateral sclerosis, commonly known as ALS or Lou Gehrig's disease, represents another target for neurological CRISPR applications. About 10% of ALS cases are caused by inherited genetic mutations, and researchers are developing CRISPR therapies that could prevent or slow the disease in these families. The approach involves knocking out the mutant genes that cause motor neurons to die, potentially preserving muscle function and extending survival. The applications extend beyond treating genetic diseases to addressing age-related neurodegeneration. Alzheimer's disease, which affects more than 6 million Americans, involves the accumulation of toxic proteins in the brain. Researchers are exploring CRISPR approaches to reduce production of these proteins or enhance the brain's ability to clear them. While these applications are still in early research stages, they represent a fundamentally new approach to treating diseases that have resisted conventional therapies for decades. The psychiatric applications of CRISPR are even more speculative, but potentially transformative. Researchers have identified genetic variants that influence susceptibility to depression, schizophrenia, and other mental health conditions. While these conditions are complex and involve multiple genetic and environmental factors, CRISPR could potentially be used to modify genes that affect neurotransmitter function or brain development. However, the ethical considerations around psychiatric applications are particularly complex. Unlike treating life-threatening genetic diseases, modifying genes that affect personality, cognition, or behavior raises profound questions about identity, autonomy, and what it means to be human. The scientific community is proceeding cautiously in this area, with extensive ethical review and public dialogue. The cardiovascular applications of CRISPR continue to expand beyond cholesterol reduction. Researchers are developing therapies for inherited cardiomyopathies, genetic conditions that cause the heart muscle to thicken or weaken. These conditions can cause sudden cardiac death in young athletes and are a leading cause of heart failure in younger patients. CRISPR offers the possibility of correcting the genetic defects that cause these conditions before irreversible heart damage occurs. Some approaches involve editing genes in heart muscle cells to restore normal protein function. Others focus on modifying genes in other tissues that produce circulating factors affecting heart function. The field of regenerative medicine is being transformed by CRISPR through the development of what researchers call universal donor cells. By editing stem cells to remove the immune markers that cause tissue rejection, scientists can create cells that can be transplanted into any patient. Without the need for immunosuppressive drugs, this approach is being applied to create universal donor organs and tissues. Researchers have used CRISPR to edit pig organs to make them compatible with human immune systems, potentially solving the critical shortage of organs available for transplantation. While xenotransplantation, transplanting organs from animals to humans, remains experimental, recent studies have shown that CRISPR-edited pig kidneys can function normally. When transplanted into brain-dead human patients, the manufacturing revolution enabled by CRISPR is creating entirely new industries. Engineered microorganisms are being used to produce everything from life-saving medications to sustainable materials. Companies are using CRISPR to create bacterial and yeast strains that can produce human insulin, growth hormone, and other biologics more efficiently and cheaply than traditional methods. The environmental applications are equally ambitious. Researchers are engineering microorganisms that can break down plastic waste, remove heavy metals from contaminated soil, and capture carbon dioxide from the atmosphere. These biological solutions could address some of the most pressing environmental challenges facing humanity. One particularly innovative application involves engineering algae to produce biofuels more efficiently. Traditional biofuel production often competes with food production for agricultural land, but engineered algae can be grown in salt water or wastewater without affecting food supplies. CRISPR modifications can optimize the metabolic pathways that produce oils and other fuel compounds, potentially making algae-based fuels competitive with fossil fuels. The space exploration applications of CRISPR are perhaps the most futuristic, but increasingly realistic. NASA and other space agencies are exploring how gene editing could help humans survive long-duration space missions and eventually colonize other planets. The challenges of space travel include radiation exposure, bone and muscle loss in zero gravity, and psychological stress from isolation. CRISPR could potentially address some of these challenges by modifying genes that affect radiation resistance, bone density, or stress responses. Some researchers have even proposed using CRISPR to engineer microorganisms that could terraform other planets by producing oxygen or breaking down toxic compounds in alien soils. The military and defense applications of CRISPR are more controversial but equally significant. The technology could be used to develop biological sensors that detect chemical or biological weapons, or to create protective treatments that enhance soldiers' resistance to biological threats. However, the same technologies could potentially be misused to create biological weapons, leading to calls for international treaties governing military applications of gene editing. The intersection of CRISPR with other emerging technologies continues to create new possibilities. The combination with nanotechnology is enabling the development of sophisticated delivery systems that can target specific cell types with unprecedented precision. Nanoparticles can be engineered to release CRISPR components only when they encounter specific disease markers, reducing off-target effects and improving therapeutic efficacy. The integration with robotics and automation is revolutionizing how CRISPR research is conducted. Automated systems can now perform thousands of gene editing experiments in parallel, dramatically accelerating the pace of discovery. These systems can test different guide RNA letters, delivery methods, and experimental conditions simultaneously, identifying optimal approaches much faster than human researchers could. The convergence with quantum computing promises even more dramatic advances. Quantum computers could potentially solve complex optimization problems in gene editing design, predicting the effects of genetic modifications with unprecedented accuracy. While quantum computers are still in early development, their potential applications to biological research could revolutionize how we approach genetic medicine. The personalized medicine applications of CRISPR are becoming increasingly sophisticated. Instead of developing one-size-fits-all treatments, researchers are creating therapies tailored to individual patients' genetic profiles. This personalized approach is particularly important for cancer treatment, where tumors can vary dramatically between patients, even when they appear similar under a microscope. CRISPR-based liquid biopsies can detect circulating tumor DNA in blood samples, allowing doctors to monitor cancer progression and treatment response in real time. This information can guide treatment decisions and identify resistance mutations before they cause treatment failure. The technology is so sensitive that it can detect a single cancer cell among millions of normal cells. The diagnostic applications of CRISPR extend far beyond cancer. Researchers have developed CRISPR-based tests for infectious diseases that can provide results in minutes rather than hours or days. These tests could be particularly valuable in resource-limited settings where traditional laboratory infrastructure isn't available. During the COVID-19 pandemic, CRISPR-based tests were developed that could detect the virus with accuracy comparable to PCR tests, but with much faster turnaround times. Similar approaches are being developed for other infectious diseases, including tuberculosis, malaria, and HIV. The agricultural applications continue to expand as researchers address increasingly complex challenges. Climate change is creating new pressures on food production, with changing temperature and precipitation patterns affecting crop yields worldwide. CRISPR is enabling the development of crops that can adapt to these changing conditions. Researchers are creating rice varieties that can survive flooding, which is becoming more common as sea levels rise and weather patterns become more extreme. They're developing wheat that can grow in saltier soils as fresh water becomes scarcer. They're engineering crops that can efficiently use nitrogen from the air, reducing the need for synthetic fertilizers that contribute to environmental pollution. The nutritional enhancement applications are addressing global health challenges. Micronutrient deficiencies affect billions of people worldwide, contributing to stunted growth, cognitive impairment, and increased susceptibility to infectious diseases. CRISPR-enhanced crops with higher levels of vitamins, minerals, and essential amino acids can help address these deficiencies. One particularly promising application involves engineering crops to produce higher levels of omega-3 fatty acids, which are essential for brain and heart health but often deficient in diets that don't include fish. Land plants typically don't produce these compounds, but CRISPR can introduce the necessary genetic pathways to create plant-based sources of these important nutrients. The livestock applications are addressing animal welfare concerns while improving productivity. Traditional animal breeding often involves practices that cause stress or discomfort to animals. CRISPR offers more humane alternatives by allowing beneficial traits to be introduced without the need for intensive selective breeding programs. Researchers have created cattle that don't grow horns, eliminating the need for the painful dehorning procedures commonly used in dairy farming. They've developed pigs that are resistant to porcini reproductive and respiratory syndrome, a viral disease that causes significant animal suffering and economic losses. They've created chickens that are resistant to avian influenza, reducing the need for mass culling during disease outbreaks. The aquaculture applications are addressing the growing demand for seafood, while reducing pressure on wild fish populations. Overfishing has depleted many wild fish stocks, but farmed fish often suffer from diseases and parasites that require antibiotics and other treatments. CRISPR-edited fish can be more resistant to diseases while growing faster and more efficiently. Researchers have created salmon that grow to market size in half the time of conventional salmon, reducing the environmental impact of fish farming. They've developed fish that are resistant to sea lice and other parasites that commonly affect farmed fish. These applications can help make aquaculture more sustainable and reduce the environmental impact of seafood production. The conservation applications of CRISPR are addressing the biodiversity crisis facing many ecosystems. Species are becoming extinct at rates not seen since the dinosaurs disappeared, largely due to human activities and climate change. While CRISPR can't bring back extinct species in the way depicted in science fiction, it can help preserve endangered species and restore damaged ecosystems. Researchers are using CRISPR to increase genetic diversity in endangered populations. By editing genes to reduce inbreeding depression, they're developing ways to make endangered species more resistant to diseases that threaten their survival. They're exploring whether gene drives can be used to eliminate invasive species that are disrupting native ecosystems. The de-extinction projects, while more speculative, represent some of the most ambitious applications of CRISPR technology. Companies like Colossal Biosciences are working to recreate approximations of extinct species like the woolly mammoth by editing the genomes of closely related living species. The goal isn't to create perfect replicas of extinct animals, but to create organisms that could fulfill similar ecological roles. The rationale for de-extinction projects extends beyond scientific curiosity. Woolly mammoths, for example, helped maintain Arctic grasslands that stored large amounts of carbon. Recreating mammoth-like elephants that could survive in Arctic conditions might help restore these ecosystems and combat climate change. However, these projects raise complex ecological and ethical questions about intervention in natural systems. The regulatory landscape for CRISPR continues to evolve as agencies worldwide develop frameworks for governing increasingly sophisticated applications. The traditional approach to drug regulation, which was designed for small molecule chemicals, doesn't always fit well with complex biological products like gene-edited cells. Regulatory agencies are developing new pathways that can accommodate the unique characteristics of CRISPR therapies while maintaining safety standards. The FDA has created specialized review teams with expertise in gene editing, and similar specialized groups are being established in Europe, Asia, and other regions. The international harmonization of CRISPR regulations remains a significant challenge. Different countries have different cultural, ethical, and scientific perspectives on genetic modification, leading to divergent regulatory approaches. This fragmentation can slow the development of new treatments and create inequities in access to approved therapies. However, international cooperation is improving through organizations like the World Health Organization, which has established advisory committees on genome editing. These groups are working to develop global standards and promote equitable access to gene editing technologies. The intellectual property landscape surrounding CRISPR continues to evolve with important implications for the availability and cost of future treatments. The fundamental patents covering CRISPR technology are owned by different institutions, depending on the jurisdiction, creating a complex web of licensing requirements for companies developing commercial applications. Recent court decisions have begun to clarify some of these patent disputes, but uncertainty remains about who owns the rights to specific applications of the technology. This uncertainty can make it difficult for companies to plan long-term development strategies and may affect their willingness to invest in certain applications. However, many companies are working around potential patent issues by developing alternative editing systems or focusing on applications that aren't covered by existing patents. The competitive landscape in gene editing is driving innovation in new technologies that may ultimately prove superior to the original CRISPR systems. The economic impact of CRISPR extends far beyond the biotechnology industry. The technology is creating new jobs in manufacturing, healthcare, agriculture, and research. Universities are expanding their genetics and bioengineering programs to train the next generation of gene editing researchers and practitioners. The investment in CRISPR research and development has reached unprecedented levels, with both government agencies and private investors funding ambitious projects. The National Institutes of Health has launched major initiatives to advance gene editing research, while venture capital firms have invested billions of dollars in CRISPR companies. This investment is paying dividends not just in terms of new treatments and technologies, but also in fundamental scientific knowledge. CRISPR research is advancing our understanding of how genes function, how diseases develop, and how biological systems work at the most fundamental level. The democratization of CRISPR technology is perhaps one of its most important long-term impacts. The relative simplicity and affordability of the technology means that researchers in developing countries can now participate in cutting-edge genetic research. This democratization is leading to innovations that might not have emerged from traditional research centers. For example, researchers in Africa are using CRISPR to develop crops that are adapted to local conditions and resistant to diseases that are specific to their regions. Scientists in Asia are applying the technology to address health challenges that are particularly prevalent in their populations. This global participation in CRISPR research is ensuring that the benefits of the technology will be more widely distributed. The educational applications of CRISPR are transforming how biology is taught at all levels, from high school to graduate school. Students can now perform gene editing experiments that would have been impossible just a few years ago, giving them hands-on experience with the tools that are reshaping medicine and biology. This educational transformation is crucial for preparing the next generation of scientists, doctors, and biotechnology professionals. As CRISPR applications become more widespread, there will be increasing demand for people who understand the technology and can apply it responsibly. The public engagement challenges surrounding CRISPR reflect broader questions about how society should govern powerful new technologies. Surveys consistently show that public support for gene editing varies significantly, depending on the specific application, with strong support for treating serious diseases but more skepticism about enhancement applications or environmental releases. This variation in public opinion underscores the importance of context-specific governance, rather than blanket approvals or prohibitions. Different applications of CRISPR may require different regulatory approaches, ethical frameworks, and public engagement strategies. The media coverage of CRISPR has played an important role in shaping public perceptions of the technology. Early coverage often focused on speculative applications like designer babies or de-extinction projects, which may have created unrealistic expectations or fears about the technology. More recent coverage is focused on proven medical applications and their real-world impacts on patients' lives. The patient advocacy communities have become increasingly important voices in discussions about CRISPR research and regulation. Organizations representing people with genetic diseases have generally been strong supporters of gene editing research, emphasizing the urgent need for effective treatments for conditions that currently have limited therapeutic options. These advocacy groups have also played important roles in ensuring that clinical trials are designed with patients' needs and perspectives in mind. Their input has led to improvements in trial design, patient support services, and communication about risks and benefits. The global health implications of CRISPR are particularly significant for addressing diseases that disproportionately affect people in developing countries. Many genetic diseases are more common in certain populations due to founder effects and consanguinity, but these populations often have limited access to advanced medical care. CRISPR technology specifically designed for global health applications could help address these disparities. Simplified delivery methods, reduce manufacturing costs, and heat-stable formulations can make gene editing treatments available in resource-limited settings. The long-term follow-up of patients who have received CRISPR therapies will be crucial for understanding the full safety and efficacy profile of these treatments. Unlike traditional drugs that are metabolized and eliminated from the body, gene editing creates permanent changes that can have effects decades later. All patients who have received CRISPR therapies are enrolled in extensive long-term follow-up studies that will monitor them for years or decades after treatment. These studies will provide crucial information about the durability of therapeutic effects and any delayed side effects that might emerge over time. The data from these long-term studies will inform the design of future CRISPR therapies and help refine safety monitoring protocols. As more patients receive gene editing treatments and longer follow-up data becomes available, our understanding of the long-term implications of genetic modification will continue to improve. Looking toward the future, the next decade promises to bring even more dramatic advances in CRISPR technology and its applications. The tools will become more precise, the delivery methods more sophisticated, and our understanding of genetics more complete. New applications that seem impossible today may become routine medical procedures, but perhaps the most important legacy of the CRISPR revolution will be the demonstration that seemingly impossible scientific challenges can be overcome through persistent, creative research. The bacterial immune systems that seemed like biological curiosities have given us the power to cure genetic diseases, improve crops, and potentially address some of humanity's greatest challenges. This transformation from basic scientific discovery to life-changing medical treatment exemplifies the best of human ingenuity and our capacity to use knowledge to reduce suffering and improve lives as we stand on the threshold of even greater advances in genetic medicine. The story of CRISPR reminds us that the most profound discoveries often come from the most unexpected places, and that curiosity-driven research can ultimately transform the world in ways we never imagined possible.